many of you in this room have or know someone with diabetes? See, that's not surprising, because globally, 1 in every 11 adults has diabetes, with 95% of them being type 2. Individuals with this disease live with a lifetime of problems, from heart disease to poor blood sugar control. And though diabetes research has made leaps and bounds in the last few years, sadly, a cure is still not within our reach. Enter MitoNEAT. MitoNEAT is an outer mitochondrial membrane protein known as a binding site to lactose, a common type 2 diabetic drug. MitoNEAT is also a metalloprotein, so it hosts an iron-sulfur cluster. Now, while the true function of MitoNEAT isn't known, previous studies have shown that MitoNEAT may play a bigger role in diabetes than previously understood. For example, when MitoNEAT was removed from gene expression in lab mice, as shown by the mouse pancake behind me, these mice remained incredibly obese, but showed little to no insulin resistance and normal blood sugar levels, subscribing to the idea of MitoNEAT's role in diabetes. So, for my thesis, we want to understand MitoNEAT's true role in the cell, besides a drug binding site, and what we have hypothesized is that MitoNEAT is in a cycle of oxidative stress. Oxidative stress, which causes DNA damage, protein oxidation, and cell death, is also a proposed cause of insulin resistance, and ultimately, type 2 diabetes. Currently, we are reacting MitoNEAT with small molecules that are products of oxidative stress, such as 4-HNE and 4-ONE. And we want to understand how this binding affects the structure and the output of MitoNEAT in cells. And what we have found is not only do 4-HNE and 4-ONE bind with MitoNEAT and change its structure, but it binds at the same amino acid that has been identified as a common binding site for MitoNEAT reactions. So, what does this mean in terms of the cycle? Now that we're identifying where these small molecules are reacting, we can hopefully find at what concentration these small molecules can release free iron from the iron-sulfur cluster into the cell. Once that free iron is released, it can react with reactive oxygen species from oxidative stress to create more of these byproducts to react with MitoNEAT and continue the cycle. We are still leaps and bounds away from a cure to diabetes. But with understanding what MitoNEAT's role in oxidative stress is, and ultimately oxidative stress's role in type 2 diabetes, we can work on that ratio of 1 in every 11 adults with diabetes and make it much, much smaller. Thank you.